Hi dear students. So today let's have a discussion on the topic autoimmune diseases. Autoimmunity basically means immunity to self, a condition that occurs when the immune system mistakenly attacks and destroys healthy body tissue. And autoimmune diseases are diseases that arise from an overactive immune response of the body against substances and tissues normally present in the body. The immune system mistakes some part of the body as a pathogen and attacks it. This may be restricted to certain organs or involve a particular tissue in different places. The treatment of autoimmune diseases is typically with immunosuppression, that is a medication which decreases the immune response. The symptoms of autoimmune diseases vary depending on the disease as well as the person's immune system. Common symptoms include anxiety or depression, blood sugar changes, digestive or gastrointestinal problems, dizziness, elevated fever and high body temperature, extreme sensitivity to cold in the hands and feet, fatigue, infertility, inflammation, irritability. Along with that, there is low or high blood pressure, malaise, weakness and stiffness in muscles and joints, weight changes, and depending on the type of autoimmune disease, there is destruction of an organ or tissue, or there is increase in the size of an organ or tissue. Autoimmune diseases almost affect every parts of the body, including brain, eyes, mouth, lungs, stomach, joints, liver, kidney, uterus, etc. Autoimmune diseases are a group of disorders in which tissue injury is caused by humoral, that is by autoantibodies, or cell-mediated immune response, that is by autoreactive T cells to self-antigens. Normally, the immune system does not attack the cell. However, there is a large group of autoimmune diseases in which the immune system does attack cells. The attack can be directed either against a very specific tissue or to a large number of tissues. Once started, autoimmune diseases are hard to stop. Let's see some of the general principles and observation about autoimmunity. Autoimmunity results from a failure or breakdown of the mechanisms normally responsible for manipulating self-tolerance in B cells, T cells, or the both. The major factors that contribute to the development of autoimmunity are genetic susceptibility and environmental triggers such as infections. Autoimmune diseases may be either systemic or organ-specific. Various effector mechanisms are responsible for tissue injury in different autoimmune diseases. Epitope spreading is also observed, that is, autoimmune reactions initiated against one self-antigens that injure tissues may result in the release and alteration of other tissue antigens, activation of lymphocytes specific for these other antigens, and exacerbation of the disease. Autoimmunity results from a failure of the mechanisms of self-tolerance in T or B cells, which may lead to an imbalance between lymphocyte activation and control mechanisms. Some of the general causes that are associated with autoimmune reactions are: the first one is the defects in deletion of T or B cells or receptor editing in B cells during the maturation of these cells in the generative lymphoid organs. Second one is the defective numbers and functions of regulatory T lymphocytes. Then there is defective apoptosis or cell death of mature self-reactive lymphocytes. Then there is inadequate function of inhibitory receptors. Then activation of antigen presenting cells, which overcomes regulatory mechanisms and results in excessive T cell activation. Now let's see some of the mechanism for induction of autoimmunity. The first one is the release of sequestered antigen, molecular mimicry, inappropriate MHC2 expression, polyclonal activation of B cells. Now let's see about the release of sequestered antigen. Sequestered antigen means the antigen which are isolated or hidden. The induction of self-tolerance in T cells results from the exposure of immature thymocytes to self-antigen and the subsequent clonal deletion of those that are self-reactive. Any tissue antigens that are sequestered from the circulation and are therefore not seen by the developing T cells in the thymus will not induce self-tolerance. Exposure of mature T cells to such normally sequestered antigens at a later time might result in their activation. For example, myelin basic protein is an example of an antigen normally sequestered from the immune system, in this case by the blood-brain barrier. Another example is the sperm, which arise late in development and are sequestered from the circulation. However, after a vasectomy, some sperm antigens are released into the circulation and can induce autoantibody formation in some men. Similarly, the release of lens protein after eye damage or of heart muscle antigens after myocardial infarction has been shown to lead on occasion to the formation of autoantibodies. The second one is the molecular mimicry or cross-reacting antigens. According to this hypothesis, self-antigens are mimicked by some non-self antigens, especially those of the viral and bacterial origin. A number of viruses and bacteria have been shown to possess antigenic determinant identical or similar to normal host cell components. Hence, the autoantibody formation induced by such molecules lead to autoimmunity. Examples of these reactions include antibodies to streptococcal antigen cross-react with some heart muscle antigens and cause rheumatic fever. Just like that, antigen of human brain and antigen of sheep brain, streptococcal M protein and heart muscles, nephrogenic strains of streptococcal antigens and renal glomeruli share similar epitopes, and all these results in cross-reaction. The third mechanism is by the inappropriate expression of class II MHC molecules. They are normally expressed only on antigen-presenting cells. The inappropriate expression of MHC2 on other cells may serve to sensitize T helper cells to peptides derived from these cells, allowing activation of B cells or T cytotoxic cells or sensitization of Th1 cells against self-antigens. For example, the pancreatic beta cells of individuals with insulin-dependent diabetes mellitus express high levels of both class 1 and class 2 MHC molecules, whereas in the case of healthy beta cells, they express lower levels of class 1 and they do not express class 2 at all. Fourth mechanism of autoimmunity is by polyclonal activation of B cells. A number of viruses and bacteria can induce non-specific polyclonal B cell activation. Gram-negative bacteria, cytomegalovirus and Epstein-Barr virus, that is EBV, are all known to be such polyclonal activators, inducing the proliferation of numerous clones of B cells that express IgM in the absence of T helper cells. If B cells reactive to self-antigens are activated by this mechanism, autoantibodies can appear. For example, during infectious mononucleosis, which is caused by EBV, a variety of autoantibodies are produced, including autoantibodies reactive to T and B cells, rheumatoid factors, and anti-nuclear antibodies. Some autoimmune diseases may have a genetic component and they are triggered by external factors such as infection or injury. Others are probably strictly caused by external factors like infection or injury alone.